Hi, this is Chapter 2, Chromosomes and Cellular Reproduction. We're going to go through some of the topics pretty quickly, as we will come back to them later in the course. On this slide, we're just touching on a review of basic cell structure and function. So make sure that what you're seeing on this screen is familiar, that that you can recall the basic structure of a prokaryote and particularly the animal and plant cell structures that are different from prokaryotes. And then read the table that shows the side-by-side comparison of some of the characteristics of prokaryotic and eukaryotic cells, just to refresh yourself a little bit. So we jump into the genetic characteristics of prokaryotes and viruses just in a very general way. You'll remember that there are two domains of prokaryotes. One's called archaea and one's called bacteria. And those do not have a nucleus, which means they don't have a nuclear envelope. They do have a chromosome, typically one large circular chromosome, but since it's not enclosed in a nucleus or in a nuclear envelope, then we say there's no nucleus. But there is a chromosome, and there are also separate, smaller, circular DNA molecules called plasmids that are found inside of prokaryotic cells. The plasmids can vary quite a bit, even between cells of the same species, and they can carry genes which are considered part of that cell's genome. In terms of viruses are fairly simple in structure. And as a general rule, all virus has some kind of genetic material, either DNA or RNA. It can be a single molecule or it can be more than one molecule. It can be linear or circular. So the genetic material in viruses varies and typically has a relationship to the type of host cell that the virus infects. There is also always a protein coat that surrounds the DNA or RNA, and that protein coat is important for the entry of the genetic material into the host cell. Some viruses also have external to the protein coat, they have another layer which is called an envelope, but it's fundamentally a phospholipid bilayer, and it can have proteins embedded in it. However, in this chapter, we're going to focus on the genetics and the chromosomes of eukaryotes. And in eukaryotes, there is a nuclear envelope. So the DNA is inside of an organelle called the nucleus. And the DNA is typically wound around histone proteins. In this image, those are shown as yellow or purpley pink structures, and the DNA is shown as sort of a green-gray string. And the DNA wraps around those histone proteins, and that material is called chromatin. The eukaryotes have linear chromosomes, but the number of chromosomes in the nucleus varies widely between different species. So we're talking about fungus, protists, plants and animals, and all of those species in those groups. The number of chromosomes it is different for most species. And then you also have extranuclear DNA. That just means other DNA in the mitochondrion, and for plants also in the chloroplasts. And that DNA is circular, very much like prokaryotic DNA. So when we look at chromosomes, linear chromosomes from a eukaryote, we will be talking about chromosomes in different forms. So on the left side of this image, you see a chromosome, and this is a double helix of DNA that's wrapped around histone proteins. And you can see that what they're calling one chromosome can also be called a monad or a single chromatid. In certain times of a cell's life, the cell will always have chromosomes in certain forms. So I gave you an example. In G1 of the cell cycle, those cells always have monad form chromosomes. But at other times in the cell's life, a single chromosome consists of two chromatids, which we call sister chromatids. They are the result of chromosome replication, and so they are identical, but they're held together in the middle at a location called the centromere. And so we say that a single chromosome, a monad chromosome on the left, is one chromosome. And then a chromosome that has two sister chromatids connected together at the centromere is also referred to as one chromosome. And that can be very confusing for students, so we'll keep working on that concept. But it's fixed. It's certain times of a cell's life when the chromosomes in that cell would always be found in the monad form, and other times in the cell's life when the chromosomes would always be found in the dyad form. So the form of the chromosomes is a function of what period of time or what phase of cell division, typically is what it is, that a cell is in. At the center, what we call the centromere of the chromosome, there are proteins that are called the kinetochore that attach to the centromere, and that's where the spindle fibers, the microtubules, are going to attach for cell division purposes. So the cell cycle is the process by which a single cell divides and produces two cells which are genetically identical. So the cell cycle consists of phases G1S and G2, which collectively are called interphase. In G1, the chromosomes in a cell are in the monad form. In S phase, that's where the chromosomes replicate. And so that's a transitional phase where the chromosomes go from being monads to being dyads. In G2, the chromosomes are always dyads. And interphase is when the cell is getting prepared for M phase. And M phase, mitosis, is defined as the nuclear division or karyokinesis, followed by the division of the cytoplasm or cytokinesis. M phase is divided into subphases that you probably recall are called prophase, prometaphase, metaphase, anaphase, and telophase. So we will briefly go over a little bit of what's happening in each of these phases, but we're focusing mainly on what the chromosomes are doing since this is genetics. So in the title of the slide, it says, make sure you understand the chronological order of the phases and subphases and the events that occur in each. And you'll find a nice summary in your textbook. Make sure you understand the purpose of this type of cell division. So this cell division produces two cells that are genetically identical to each other, but that's not the purpose exactly. The reason why an organism would want to make cells that are genetically identical is for growth of the organism overall. So when you're a child, you have fewer cells in your body than an adult. So by having this kind of cell division, we have the organism is growing. But also later in life, cells will go through this division to replace other cells that have died or to help with repair processes of tissues. So we say that the purpose of the cell cycle is to produce genetically identical cells for the purpose of organism growth, tissue repair, and maintenance. So I'm going to go through and fill in what the chromosome form is at each stage of the cell cycle. And to do that, I'm going to go back into edit mode. This is the picture on the right side here of the cell cycle, specifically focusing on prophase, metaphase, anaphase, and telophase. And we're going to fill in this chart for what form the chromosomes are in, which you can, for the most part, see in the diagram. And notice how the diagram goes from left to right, but then as you go down, you go from right to left, and you end at telophase. So in G1, you can't see the chromosomes well with a microscope because they're not condensed. But in G1, the chromosomes are in the monad form, in the single chromatid form. And the number of chromatids per cell is whatever the diploid number is for that species. In S phase, that's when the monad chromosomes transition to the dyad form because of DNA replication, but you still say that you have a diploid number of chromosomes in the cell. So remember that the centromere, there's one centromere in a monad chromosome, and there's one centromere for a dyad chromosome, and we count the centromeres in order to count the chromosomes in the cell. And that's why the number of chromosomes would not be said to change. In G2, the chromosomes are still in their dyad form, following their replication in S phase, and they stay in the dyad form until prophase and through metaphase. Then there's a weird moment, I guess you can say, in anaphase. Remember, when the dyad chromosomes each are pulled apart into their separate sister chromatids, we say that this is a transition from the dyad form back to the monad form. So, and briefly, in anaphase, we would have to count each of those monad chromosomes. So briefly, the cell would have twice whatever the diploid number of chromosomes was. In telophase, the chromosomes are still in the monad form, and each newly forming nucleus has a diploid number of chromosomes, but the entire cell, if you counted the whole cell prior to cytokinesis, it would still have twice the diploid number. But then following cytokinesis, you have two cells, each containing monad chromosomes and each having a diploid number, and that cell is returned back into G1, and then the whole thing repeats.